let's get started with our first topic this year, which is going to be physiology. It is topic 6 in the IB guide, and it'll be our unit 1. So we're going to start off with 6.1, which is the digestive system. This is an intestinal tract camera. So this is a way that we can see what's going on inside someone's body if they have a blockage or just in general to do research. It's very, very small, as you can see by the measurement below. And you could swallow it easily and we could see what's going on inside you. This is an image of the digestive system and the organs that make it up. So it's a system, right? And you need all these organs to make this organ system. So if I were to ask you to label a diagram, these would be the important ones that you need to remember. So starting with the mouth, you can see that this is where the food would enter, going down the esophagus to the stomach. Then it would go into the small intestine here. And then eventually it's going to go into the large intestine and then out the anus. There are some organs that are a part of the digestion process, like the pancreas, the gallbladder, which are going to help send enzymes to break down those polymers into monomers. And then they're eventually going to be reabsorbed into the liver to be used later for our body to make us be healthy and active and recreate some of the breakage that's maybe happened in our body and re-up on some of those nutrients we need. So if I were to show you a real life digestive system, this is a part of it. So this is a pig, a fetal pig, which you guys will be doing a dissection for a fetal pig at the end of the year. But what you can see is it's all packed together really tight in our bodies. And our digestive system is very similar to the pigs because we're both mammals. And you can see this is the liver here, which actually covers up a lot of the area. So this is the liver here that absorbed all those nutrients. Here's the stomach under the liver. It's smaller than you would think. Here's the large intestine. And then here is the small intestine. There's a lot of it all pushed together. The small intestine, there's probably the most of it. You can't even see the pancreas in this image because it's behind further the stomach. So this would be an idea of what it looks like, and it's kind of hard to tell until you pull it all apart and stretch it out, but you can see that there's a lot all packed into one space. So in order to get into the digestive system, we have to go back to our unit 2 from last year, which was about biochemistry or molecular biology. So what we talked about then, remember, was macromolecules, so proteins, lipids, nucleic acids, and carbohydrates. Those are the 4 big macromolecules. And we talked about how through different processes, for example, hydrolysis, you can break apart polymers into monomers, or you could go back the other way and build polymers from monomers by adding water. In this case, water was added and it broke it apart, or by taking water out. So if we remove those, they would go together. So macromolecules are substrates in the sense of digestion, and then we use enzymes to break them apart. So we're going to review the idea of enzymes that we also talked about from last year. Remember, enzymes are a type of protein, which is a macromolecule. So here's just a little summary of condensation reactions versus hydrolysis reactions. So you can see in red, it's when you add water, which would be a condensation reaction. And what you can see is when water goes in, you break it apart. Okay, or I'm sorry, the red line is the hydrolysis reaction. So when water goes in, it's going to break it apart. And a condensation reaction is when water goes out. So think of like evaporation or when things condensate. Water goes out, and that's going to bond them together. So that's going to create a chain or a polymer of those monomers. So this would be an example of a protein. So just kind of getting used to seeing the structures again. So remember, the monomer for a protein is an amino acid, and the polymer is a polypeptide. So this would be 2 protein or 2 amino acids together, and this would be more than 2, which makes a polypeptide. And you can see on the top is the hydrolysis reaction, which is breaking it down into a monomer. And on the bottom in blue is the condensation reaction, which is putting it together and making a chain or a polymer. Down here is the carbohydrate. So you can see, remember, this is a monosaccharide, which would be a a glucose molecule. And if you were to put them together, you'd get a disaccharide, and then a longer chain than 2 would be a polysaccharide. So you can see 3 of them bonded together. If it was a hydrolysis reaction, you're breaking it down to the original monomer. And if it was a condensation reaction, you'd be putting it together into a polymer. So this is what's happening in our bodies when we digest food. We are usually breaking things down to monomers, and then later on, our liver is going to put them back together into a polymer for us. So looking at human digestive enzymes, so these enzymes that are going to break down our food for us into monomers, these are just a few of the important ones. Amylase, which usually is going to break down the carbohydrate, so the macromolecule. An example would be salivary amylase, which is in our mouth for saliva. And it's going to break down the first things that enter our mouth, which is usually a carbohydrate or starch that is paired with other ingredients as well, like proteins, but like the breads or anything that has a sugar in it. Like a fruit has lots of sugar in it, so it has lots of carbohydrates. So it's going to start breaking that down, that starch, which is the substrate. The product, the monomer at the end, is going to be maltose. And then the source where this is happening in our body is our mouth, our salivary glands. And the pH that's making this enzyme be able to work, because remember, enzymes need an optimal condition or environment, would be that our mouth sits at a pH of about 7 to 7.8, so slightly basic. Okay. The question down here says, where might we find enzymes with an optimum pH of 2? Give you a moment to think about it. Which part of our digestive system or organ do you think has a very acidic environment, right? pH of 2. And maybe you guessed it, it's the stomach. So the stomach is going to be where we see, because of that pH, it allows the enzyme pepsin, which really needs acidic conditions to work, to break down the proteins in our food. Here's another one, protease. You need to know these enzymes. There's about 4 or 5 key enzymes you need to know, and you need to know what macromolecules they break down. So protease breaks down proteins. An example of a protease is trypsin. Its substrate is a polypeptide, so a chain of amino acids, right, a bigger protein, and it's going to break it down into amino acids, the monomer. This usually happens in the small intestine, and it's the beginning of the small intestine. That's called the duodenum, and the pH of it is an 8 in the environment, and that is what this enzyme protease works best at. Lipase, you guessed it, breaks down lipids or fats. So pancreatic lipase, so it's released by the pancreas, which so is trypsin. So you'll notice a lot of these enzymes are released at the pancreas, except this salivary amylase. There's also another amylase that is released by the pancreas as well. So it breaks down triglycerides into fatty acids and glycerol. So remember, one lipid is usually a fatty acid chain with a glycerol head. So this happens in the pancreas, and it works best at a 7.2, 7.5 pH, the enzyme does. So this is a picture up here, this purple dot, of a substrate, and these are the enzymes. Remember, every substrate has an enzyme that fits perfectly with it, right? There's like a lock and key method, and it initiates it to start breaking down whatever that substrate is. Okay, so this is going through the pathway, so follow the numbers of digestion. So what happens in digestion? First, it starts with the mouth. So chewing, which we would call mechanical digestion, not chemical digestion, because it's working with the muscles that are trying to break it down. Saliva is going to be released, which is going to contain amylase, which will begin to break down those carbohydrates. Then it's going to go down the esophagus, which will then head to our stomach. The esophagus is here. So it's a wave of muscles that with the contractions will help push it down, and the food will have turned into this bolus, this ball, and it'll push it down to our stomach. The stomach is going to also do mechanical digestion because it's basically a bunch of muscles, and it's going to do contractions to kind of break down that food. There are bacteria that live in our stomach, and they release the bacteria that are on the food are going to be killed by the acidic condition so that we don't get sick, hopefully, by any bacteria we've consumed. And then also the enzyme pepsin is going to be released. And remember, it works best in an acidic environment. And it's going to begin working on the substrate of proteins, okay, so the polypeptide chains. After it goes through the stomach, it's going to work itself down into the small intestine. The beginning is called the duodenum. So that's over here, the small intestine. And there's a lot that's going to happen in the duodenum. So this is where all the enzymes that the pancreas releases is going to release them to this point. So also the gallbladder is going to release bile from the liver. And this is going to neutralize the acid and emulsify fats. We call them bile salts. So they basically coat those fats because remember, lipids are oily and they don't mix with water well. So if we add this salt-like material to them, it helps neutralize them so that we can break them down. So the gallbladder is going to release into the duodenum bile salts. And then the pancreas is going to release into the duodenum amylase to help break down carbohydrates again, lipase to help break down the lipids that are there and fats, trypsin, which will help digest proteins, as well as other things like chromatrypsin will be there. That'll help break down proteins. And nuclease, which isn't in this picture, which will help break down any nucleic acids, the fourth kind of macromolecule. Once things have broken down further, they're going to move down through the small intestines. And this is where, so step 5, the ileum, this is where nutrients are going to be absorbed into the body. So this is where we get back from what we've digested. So there's these little villi that increase the surface area so that there's lots of surface for these nutrients to be absorbed. And once those nutrients are absorbed, they're going to head to the liver for the liver to start trying to put them back into polymer chains for us to use later on. Whatever is remaining after the small intestine from what was not absorbed is going to go into the large intestine. This little thing hanging off the bottom is the appendix, which we as humans no longer have a use for, but other animals do. So it's going to move through the large intestine, and then it'll eventually come out the rectum, or it'll go to the rectum and then through the anus. So the main thing about the large intestine is this is where water is going to be reclaimed to help us with staying hydrated. And it's going to return it to the blood, and it's going to leave then just a semi-solid feces. So at the end, that can then be released by the anus. So feces contain undigested food, dead cells, and other waste, and it's forced out the anus through contraction. So this is a very important slide. You're going to refer back to this a lot when trying to understand the pathway and what enzymes are used. So just to kind of get into the stomach a bit more, if you're looking at it, you can see it's made of the muscles mostly. So it stores and mixes food, aiding in both physical and chemical digestion. HCl, or acid, produces a pH of about 2, facilitating pepsin activity, the enzyme pepsin, in killing any bacteria. Pepsin, remember, is going to catalyze the hydrolysis of proteins into smaller polypeptides, so amino acids. Chyme is also a product of the stomach digestion. It's an acidic fluid released from the stomach into the small intestine via pyloric sphincter, which is right here, which is going to help bring down, that'll be the next stage, this chyme that's going into the small intestine to be broken down. So this one's about the small intestine, which is the most important here. So you can see we have the duodenum up at the top, and then as you move into the ileum in here, okay, there's the duodenum and then the ileum, but you just really need to remember it all together as a small intestine and the duodenum in the beginning. So its muscles in the wall of the small intestines contract to mix the food with the enzymes. The pancreas is going to release bicarbonate, which is going to neutralize that chyme because it's still a little acidic. So remember, if you mix a basic and an acidic condition, it'll help neutralize it and bring it more towards a pH of 7. This will produce a pH of 8, optimizing the activities for the other enzymes that are going to come from the pancreas. Here is the list of those that you need to know. The amylase, which will break down carbohydrates. protease, which is going to break down proteins, lipase, which is going to break down fats, and endopeptidase, which is going to break down peptide bonds between amino acids. The one that's not mentioned here is the nuclease, which will break down nucleic acids, which are part of our DNA, or anything we eat's DNA. So bile is then produced in the liver, remember, and it's stored in the gallbladder, and this will help be released into the duodenum to help emulsify the fats, which can then be broken down further because they're lipids and they don't mix with water well. So the liver is going to release these bile salts, or the gallbladder is going to release these bile salts, but the bile is produced from the liver. This is what the surface of the small intestine looks like. So notice all these little fingers that are coming off of the small intestine. This creates lots of surface area. They are all a bunch of little cells. Think of all of these as these little black dots or nucleuses all along the edge of the small intestine's lining. So that means food can come in, or nutrients can be absorbed into the bloodstream through all of these little villi, and then they'll eventually pass to a capillary that's in here, which will take those nutrients to the bloodstream and send them to the liver. So because of this structure, remember, structure determines function, this allows so much absorption to be happening in the small intestine. So just to see it a little bit differently, remember when we talked about surface area, the larger something is, the harder it is to absorb everything to the center, right? So if there's a lot of little holes or little curves, like in this case, the villi have, it creates more surface area for it to absorb. So an increased surface area is going to let more nutrients get in more efficiently to these red capillaries, which will then take the blood, the nutrients through the bloodstream to the liver. So you can see here even smaller, these little fingers on this side, and then this is zoomed in on just one of these little fingers here. So we zoom in and you can see all these little villi. So folding is going to increase the surface area by 3 times. The villi within each fold is a second set of folds, creating a series of villi. So just having these folds in general is important, but then within each fold, there's even more. So that's going to increase it by 10 times. The microvilli along the lumen side of each small intestine epithelial cell brush a border of microvilli additionally, which will expand the surface area by 200 times. Thus, the total surface area increase up to 600 times. So just one thing you have to understand is that there is a different method for getting these nutrients depending on what they are across the villi. So different types of diffusion that has to occur. So a single layer of epithelial cells allows for fast diffusion. Diffusion of fatty acids, monoglycerides, fat-soluble vitamins, and some mineral ions are going to happen through the phospholipid bilayer. So remember when we talked about the bilayer of a cell, the phospholipid bilayer, they have to be diffused through that. Facilitated diffusion of some monosaccharides and some vitamins and mineral ions like calcium using membrane proteins. So they're going to be going through the proteins that go through that phospholipid bilayer. And then active transport of amino acids, and most monosaccharides, some mineral ions using membrane proteins, and ATP produced by mitochondria in the epithelial cells. So some require active transport, so energy is needed to open up the channel so that they can go through. So sometimes this has to do with size of the molecule, and sometimes it has to do with the polarization of the phospholipid bilayer. Right? So it's hard to get things through the center that are polar because the center of the phospholipid bilayer here is nonpolar. So realize there are different ways to diffuse through the phospholipid bilayers of these villi cells, right? Because they each have a phospholipid bilayer. So this is giving just a more close look. So you can see here are the villi, and then they each have little projections of fingers around each called microvilli. The lacetal is here in the center. The lacetal has many small vessels of lymphatic systems, which carries clear watery fluid containing a protein molecules, salt, glucose, and urea. Absorption of lipids, which are too big to pass through the blood capillaries, will happen here in the lacellate. But if they're small enough, they can pass through the red and blue sections here. They can pass into the bloodstream this way. Those would be the blood capillaries. And then notice they're going to come down into these tubes to move through to larger vein systems. So capillaries lead to the veins. You have to be able to, for a test, tell when you're looking at an image of the small intestine. And what gives that away is you can see, here's the muscle layer, but then inside you can see the villi. You can see these large finger-like projections, and that's what gives it away. So this is the skill here, you can see. So identification of tissue layers in a transverse section of the small intestine viewed by a microscope or a micrograph. So these are from a micrograph. So the small intestine contains 4 distinct tissue layers of the lumen. So the mucosa, which is the inner lining, including the villi. Then the submucosa, which is the connective tissue between the mucosa and the muscle. So you can see this is the muscle layer here, this thick part. Then here's the submucosa here that's connecting to the mucosa, which is containing all of the inner lining and the villi. Here's a larger image of it to zoom in. You can see the muscles here for contraction to help keep moving things along. The submucosa here, the mucosa that contains all of the lining and the villi. And the lumen, remember, is an opening. Lumen just means opening, so there's a hole in the middle here. So absorption and assimilation. Digestion is breaking down the large food molecules. So that's one part of the digestive system. But the second part of the digestive system is absorbing it. We want to get from those nutrients. We don't want to lose it, right? We don't want it all to release from our body. So absorption is the uptake of these molecules into the bloodstream. So once in the blood, they're carried to the tissues where they are assimilated or taken in to be used. So that's why the small intestine, besides the digestive process, is super important for further on down in the small intestine, where we are going to be absorbing these nutrients. So that's why those villi are so important that we talk about them, because either we will absorb through the cells into the capillary system, or we'll absorb here into the lacetal. And either way, we're going to take all those nutrients back to the liver to assimilate them and use them. So the liver, when there is a lot of sugar in the blood, insulin is going to be released. Okay, so it's triggered that way. Hepatocytes, which are liver cells, will also use enzymes to add monosaccharides to the long chains of glycogen stored in the liver. When energy is then needed, it'll signal to the liver, our body will, and glycogen will then be broken down into glucose again so we can use it. So the liver is building up those glycogen chains from what's been absorbed to then be used when we need to make or to use energy. Okay. So then to the large intestine, it's where there's a reabsorption of water occurring, as well as sodium and potassium from the intestinal lumen to the capillaries. So there is still this exchange or absorption happening between a capillary system of those 3 things. So the movement's happening by peristalsis, which is a rhythmic contraction of these circular and longitudinal muscles. And the reason why it's circular and longitudinal is one muscle is moving more sideways and one's moving straight up and down. And this movement together is helping it keep moving forward. And it'll eventually slowly make its way down the intestinal tract as a ball of feces. So that's the end of our lecture. And we are going to be doing a skill. After listening to this lecture, I want you to take notes, and I want you to put them into ManageBack, okay? We're doing this activity a different day. So watch the video, take notes, and upload it to ManageBack as a PDF or Word doc. Thank you for listening.